Well, hello there again. This is video number 2, or the second video in our discussion of cell structure and function. And again, here I'm going to begin by pointing out this link to a YouTube video, which essentially is a tour of the cell. You can't watch the video through this video. However, what you can do is download the PowerPoint from Canvas and click on this link in the PowerPoint, and that'll give you an opportunity to watch. All right, just a different perspective on the various things that we're going to talk about in this video. Now, so we'll start today with a discussion of what a eukaryotic cell looks like. Remember, there are two major kinds of cells, prokaryotic and eukaryotic. The prokaryotic cells are very simple, very tiny little cells that, generally speaking, have no real organelles inside of them. Eukaryotic cells are much larger, up to 20 times larger than a prokaryotic cell, and they're very complex. Now, the picture that you see here is just an illustration of what eukaryotic cells actually look like. And you can see that they're incredibly complex, all right? And remember that each of the cells has to carry out each of the life processes, all right? All of the things that we talked about in that very first section there. And so it is important that you understand not only the structure of the cell, but the function of each of those structures. Now, we'll start at the outside of the cell and we'll work our way into or toward the middle. Now, this is a cartoon representation of what we call the plasma membrane. All right, your high school biology teacher may have called it the cell membrane. Same thing, okay? It is a phospholipid bilayer. Now, if you remember from chapter 2, a phospholipid is a kind of lipid, but one of the 3 fatty acids in that lipid has been replaced by a phosphate. All right. Now, this gives a phospholipid some interesting characteristics. All right. The whoopsie daisy. This little this little balloon-like looking thing is the head. This is the glycerol. And these 2 little tails that you see here are the fatty acids. Now, one of the fatty acids has been removed and replaced with a phosphate, which gives the head a hydrophilic or water-loving ability and makes the tails, the fatty acids, hydrophobic, which means that they hate water. And so what happens is you end up with this bilayer where the area in here is all made of hydrophobic or water-fearing molecules, and the top and the bottom represent hydrophilic layers, which are water-loving. Now, notice that inside the picture, there are these brown things. All right, look, this one looks almost like a hot dog bun. But those represents proteins that are embedded within the phospholipid bilayer. And there are some other things in there as well, some carbohydrates, some lipids, and so on. Now, 2 things that are important about the plasma membrane. Number 1, it is a fluid membrane. In other words, it's not completely solid. In other words, if you choose this little phospholipid right there, in 10 minutes, it might be over here. All right, and this one is going to go over here, and this one is going to go over here, and so on. They're constantly in motion. All right, and so it's not literally a solid brick wall, if you will. This allows it to be what we call selectively permeable, meaning that it will allow certain molecules in and certain molecules out. All right. It will also prevent certain molecules from getting in or out, depending upon their structure. All right. More on that in a little bit, while. All right. But keep that in mind. Selective permeability is kind of like why we put a screen in our window during the summertime, right? We open the window because we want the fresh air to come in, but we don't want everything that's outside to also come in, right? We don't want the bugs and the birds and the bats and whatever else. So we put a screen on the window that lets the air in, but keeps the unwanted stuff out, all right? So a screen is selectively permeable based on size, all right? Small things like air molecules can get in and out. Big things like bugs can't get in and out. Okay, so what exactly does selectively permeable mean? All right, well, we just kind of talked about that. It means that the membrane will allow certain things in while keeping other things out. And this diagram illustrates the four real classes of molecules that are important here. Now, if you are a small molecule that does not have a charge, you get in and out very freely. All right. So things like air, oxygen, carbon dioxide. All right. Even water molecules to some degree, although there are special channels that water can get in and out through. But these things will diffuse right through that membrane very, very easily. On the other hand, charged molecules, things with a positive charge or a negative charge, are actually repelled by the tails, the hydrophobic tails of these phospholipids, and they're kept. You can see here, all right, they're kept from going in or getting out of the cell. And then finally, there are macromolecules. Now, this thing represents a protein of some sort, but this macromolecule is simply too big to squeeze between the phospholipids in the plasma membrane. And so they are prevented from getting either in or out of the cell. Right? So cell membranes are selectively permeable. Okay. Now, it doesn't help a cell at all to have a solid membrane because then things can't get in and out. But what processes do we use to actually get things in and out? Well, we've already mentioned the idea of diffusion, okay? And by definition, diffusion is the random movement of molecules from a high concentration to a low concentration, all right? This is a time-lapse cartoon, if you will. And you can see that on the outside here, you've got lots of water molecules, and on the inside there are there are fewer water molecules here and here. Okay, there are also some particles. This could be a salt. It could be some ion of some sort. It can be a lot of different things. But over time, why does it keep going backwards? Over time, you'll notice that eventually we reach equilibrium. The water molecules diffuse into the cell, and so do the salts, or whatever these little purple particles are supposed to represent. Now, osmosis is the diffusion of water molecules. And so diffusion can be a little bit counterintuitive, because what happens is that the concentration of water is high when there's very little other stuff dissolved in it. And the concentration of water is low when there's a lot of other stuff dissolved in it. And so water tends to move in the opposite direction as to what we generally think. But if you just look at the concentration of the water molecules themselves, you can see high concentration beforehand on the outside, and therefore the water will diffuse into the cell. I have a very sensitive mouse, apparently. It doesn't seem to want to pay attention to what I want. Anyway, so diffusion and osmosis. Now, what's important about diffusion and osmosis is that neither one of them requires any energy at all. All right, so this is a free transport of materials in and out of a cell. Now, what do we do if there are molecules that we want to move across the cell membrane, but they're too big, or they have a charge and they can't diffuse through that phospholipid bilayer. Well, in that case, we can do one of two things. First, there's such a thing called facilitated transport, all right, which is sometimes referred to as passive transport. And this is the transport of molecules across the plasma membrane, again, from higher concentration to a lower concentration, but using a protein carrier, all right? This is shown here. This is a transport protein that's bringing a glucose molecule from the outside of the cell to the inside of the cell. And essentially, it's just kind of like a passageway, a tunnel, if you will, a gate that doesn't require any energy to be expended by the cell itself. Now, sometimes, no matter what you do, you still can't get stuff to where you want it. All right. And in that case, you need to revert or rely on what we call active transport. Now, this is different from the other three because it requires energy. The cell has to expend energy in order for this to occur. This is the movement of molecules, now get this, from a lower concentration to a higher concentration. So we're going against the concentration gradient. And we have to use the energy carrier molecule, ATP, in order to make this work. This is illustrated on the right side of lay screen. And this one represents what we call the sodium-potassium pump. It requires a molecule of ATP. One of the phosphate bonds is broken, and the protein structure literally changes shape. Remember, we said when we were talking about proteins that their shape determines their function. So what happens is the protein is shaped like this, all right? A couple of well, actually, it starts out in this way. A couple of potassiums come in and bond to it, and that causes the whole shape of the protein to change. That releases the potassiums to the inside of the cell and then allows sodiums to be carried to the outside of the cell. But again, the change in the conformation of the protein from back and forth requires energy, requires ATP. And so therefore it's called active transport, whereas the other three are passive. They don't require energy at all. So this brings us to the third item on our list of important things. All right, remember that this is a list of things that are going to be important for your final exam. So you've already got 3 questions for your final exam laid out for you right here. Diffusion is defined as the movement of materials from an area of high concentration to an area of low concentration without using energy. All right, the movement of materials from an area of high concentration to an area of low concentration without using any energy. All right, so diffusion, osmosis, and facilitated transport are all forms of diffusion. Active transport is obviously not, all right, because we're going against the concentration gradient there. Okay, now at this point, let's talk a little bit about these insides of the cells. Now, the nucleus is, and you can see, here's the whole eukaryotic cell. This big purple thing represented here is the nucleus. All right. The nucleus itself is bound by its own membrane, also a phospholipid bilayer, just like the plasma membrane. And inside the nucleus, you find the DNA, the genetic blueprint for the cell. In addition, inside you find some proteins that help to hold the DNA together, and the DNA and proteins together are a material we call chromatin. It's got its own fluid inside that we call nucleoplasm, and a very dense region right in, well, it's not necessarily in the middle, but inside the nucleus that contains a special form of RNA called ribosomal RNA. We'll get to that in just a minute. Now, so the nucleus is surrounded by the membrane, and if you look very carefully here and here, you'll notice that there are little pores, and these are designed to let messenger RNA out of the nucleus and other things into the nucleus. So diffusion can take place through those little pores. Now, just outside the nucleus, you see all of these teeny weeny little yellow dots. There's one right there, okay? And these are organelles, little organs, literally, that are made of RNA and protein. And oftentimes, they're found bound to another organelle called the endoplasmic reticulum, but we'll come back to that later. And you can see them all here. All right, here they are, all these little yellow dots, and this this weird membrane that goes back and forth here is the endoplasmic reticulum. The ribosomes are important because they are the sites of protein synthesis. This is where the amino acids are strung together in that first long chain, and then we either make an alpha helix out of them or the beta pleated sheet. But we'll get to the endoplasmic reticulum a little bit later on, okay? Now, proteins are exceptionally important in the body for a number of structural and functional reasons. And at this point, I want to take a step away from the cell and actually talk about what an enzyme is, all right? Enzymes are, first of all, very important for cellular respiration and for many other metabolic activities inside the cell. They help to guide various chemicals together, and they also help to take certain chemicals apart. And most enzymes are proteins, which is kind of why we talk about it when we're talking about ribosomes. Now, the enzymes are often named after the molecule that they work on. All right. For example, there's an enzyme called lactase. All right, and that is an enzyme that's designed to break down the milk sugar lactose. All right, and so they have similar names. The ASE is typically the enzyme. OSE is typically a sugar. And so there's lots of different kinds of enzymes that are important in human biology. You've got proteases, which help to either break down or put together proteins. You've got amylases, which are important in breaking down carbohydrates. And you've also got lipases, which are important in breaking down lipids. Now, enzymes are very specific to the substrate that they work on. All right, it functions just like a lock and a key. And as I said the other day in the last video, you cannot put your car key into the lock on your dorm room and expect that it's going to work. You can't put your house key into your car ignition and expect that it's going to work. All right, keys are very specific to the locks that they open, and enzymes are very specific to the substrate that they work on. The structure of an enzyme, because it's a protein, all right, the shape of the protein is exceptionally important. And there's a special place in that protein that's called the active site. The active site itself is designed to fit into a specific protein. So this enzyme here is designed for a hydrolysis reaction, for example. You take a substrate molecule, and notice it plugs right into the active site on the enzyme, and here it is. And then the products are then released. All right, and so we've broken this molecule down into two smaller molecules. Notice also that the enzyme is unfazed by this. All right, once the chemical reaction has taken place, the enzyme is free to go and do it again and again and again. And most enzymes can be used hundreds or even thousands of times before they degrade, they break themselves down. On the other side of the screen is a synthesis reaction. Now, this is obviously a very similar kind of thing. We take the two substrates, we plug them into the active site, and we bind them together to form the product here. And again, the enzyme is recycled. Now, these two enzymes are different, all right? The one that breaks the molecule down is different from the one that puts the two molecules back together to make the original molecule, all right? but they have similar active sites and similar jobs. Now, what is the endomembrane system? Well, this is a series of membranes in which molecules are transported throughout the cell. There are several major parts of the endomembrane system. It starts with the nucleus and the nuclear membrane and leads into the endoplasmic reticula. From the endoplasmic reticula, we go to the Golgi apparatus or the Golgi bodies. And then there are little membrane-bound sacs called vesicles, which carry stuff. And then lysosomes, which are specialized vesicles that carry digestive enzymes. Now, let's go through each of these, and then we'll talk about how they might work. The endoplasmic reticulum is divided into two different pieces. The rough endoplasmic reticulum is studded with ribosomes, all right, which gives it the rough look under a microscope. And the ribosomes, remember, are the protein factories. They are the site of protein synthesis. Smooth endoplasmic reticulum is similar, but it doesn't have the ribosomes, and so therefore it's not being used to make proteins. But the smooth endoplasmic reticulum is very important in creating lipids. All right, it says carbohydrates and lipids on this slide, but in reality, lipids are the main function there. The Golgi apparatus are flattened stacks that process, package, and deliver proteins and lipids from the endoplasmic reticulum. All right, so things are shipped from the endoplasmic reticulum to the Golgi apparatus and then off into Never Never Land. It's kind of like the Golgi apparatus is kind of like the UPS store. All right, stuff comes in, the UPS guys package it up, slap a shipping label on it, and send it on its way. All right. The vesicles are the transport sacs. They kind of like the trucks that the UPS drivers use. And lysosomes are specialized vesicles that contain digestive enzymes. So let me show you what this might look like. All right, we start here at the nucleus, and a little piece of RNA is sent out into the rough ER, and this area here is the rough ER. And the RNA tells the ribosomes what protein to produce. Then, come on, a little piece of the end of the endoplasmic reticulum pinches off and becomes a vesicle. And this transport vesicle carries the proteins up to the Golgi apparatus. That's this thing up here. And here, the proteins are taken in. They are rearranged a little bit. They are then repackaged into a new vesicle with a shipping label on it, and it's then either sent out of the cell or to another place within the cell in a secretory vessel, all right, for secretion. The same thing is going on in this picture, but over here is the smooth ER. And instead of producing proteins, we're producing lipids. They're packaged into small vesicles, which again go up to the Golgi bodies, where they're repackaged and attached to a shipping label and then sent off into wherever their destination happens to be. The last thing that I want to point out is specialized vesicles called lysosomes, and this is represented by this picture here. If a large molecule is taken into the plasma membrane and surrounded by a vesicle, that vesicle may meet up with a lysosome, and the digestive enzymes will then break whatever that molecule is up into smaller building block molecules. Okay. The last organelle that I want to talk about is the mitochondria. All right, and mitochondria are small organelles. They are highly folded. And why do you think they might be highly folded? Well, that increases the surface area inside the organelle. As a matter of fact, if you go back here, why do you think the Golgi bodies and the endoplasmic reticulum are all flattened out? because that increases the amount of surface area that's available for the metabolic reactions to take place on. In any event, the purpose of the mitochondria is to produce energy in the form of adenosine triphosphate, or ATP. And so the more membranes they have, the greater the surface area that's available to produce that energy. Now, interestingly enough, the mitochondria are thought to have evolved from prokaryotic cells that were engulfed by bigger cells. And there's a couple of reasons for this, but mainly it's because mitochondria actually contain their own DNA that's not part of our genome. It doesn't contribute to our genetic genotype and phenotype. Now, and they also reproduce independently from the rest of the cell. Now, inside the mitochondria is the process, or occurs the process of cellular respiration, which is where we take glucose and oxygen and produce ATP. And this, we don't need to go through all of the steps in the chemical processes, but there are three major steps: glycolysis, citric acid cycle, and electron transport chain. But in the end, one molecule of glucose that's brought into the cell and sent to a mitochondrion will end up producing around 36 molecules of ATP using oxygen and the glucose that we're sent in with. All right, so that is the process of cellular respiration. The oxygen comes in. What's the waste product? Carbon dioxide. Okay, the carbon dioxide is released actually from the citric acid cycle part of this. All right, boys and girls, that concludes our discussion of chapter 3. I thought I was going to do this in 3 videos, but I ended up doing it in 2. So I hope this one wasn't too long for you. Have a great day.